Awesome. I think it's
Okay.
working now. Go
Is it
ahead.
okay for
Thank
you?
you.
You get
Yes,
it? No?
it's in full screen mode. You may go ahead.
Okay. Thank you. We can finally make it. So, hi everyone. I would like to thank you for inviting me for this cozy pathogen webinar. I will talk about part of part of my interest on this bioinformatic field. So. My name, first of all, my name is Osama Suyai. I am a computational biologist, bioinformatician, lecturer at the University Tunis Manar, and I'm research associate in the Pasteur Institute of Tunis and part of H3 Bionet node, Tunisian node. So this the today's talk will be about the delivery mode impact on pathogenic species prevalence through microbiome and microbiome analysis. So just to give you some insights about our projects, the projects that we are working on the laboratory, we have microbirth, which is the interest of today, and dealing with the impact of delivery mode on pathogenic prevalence in newborns microbiota. And we have also some projects related to genomic characterization of various microbial pathogens. So um,
I don't know if it's starting again. Dr. Sawaya, we can't hear you. I think your audio has gone. Okay, I'm just sending. Oh, okay.
okay. Sorry, did I lost you?
Yes, on the slide, I think it was the previous one.
This one.
Yeah.
So the first
The
one. No?
previous one.
Okay, I will start. So it's told that the acquisition of and the development of the gut microbiota occurs during early life. The first days and weeks of life represent a crucial and very determinant window of opportunity for shaping the development of the gastrointestinal tract and the immune system. This stage sets also the basis, for the adult microbiome.
Dr. Sawaya, are you using headphones or earphones? Because perhaps you're going to try it without the headphones, just using your laptop's audio, because I think we lost you again there. Okay, my apologies, everyone. I think there's a bad connection. At this stage, we're not sure if it's the internet connection or just audio problems. So we're just going to hang in there and wait for Dr. Sawaya to connect again. In the meantime, maybe we can. Oh, he's back. Okay.
Sorry. Okay, can you hear me again?
Yes, we can. Thank you.
Okay. What I wanted to say is that therefore it's important to deeply understand the factors that shapes pioneer microbes to properly provide a new perspective for the prevention and treatment in case of disease. The composition and development of infant gut microbiota can be influenced by many prenatal and mother-related factors, such as not limited to maternal diet, obesity, smoking status, and use of antibiotic agents during pregnancy. But following birth, it was well established that feeding practice, antibiotics use, and the surrounding environment are the major factors that shape the initial composition of newborn's gut microbiota. However, the real impact of delivery mode is not fully resolved and still subject to many debates. So, indeed, it was widely accepted that, first, that infants delivered by cesarean section miss contact with the maternal vaginal microbiome, and this will lead to an unbalanced and disequilibrated early establishment and development of microbiome. This immature microbiome in CS infants therefore lacks diversity, and this would lead to adverse later life outcomes such immune and metabolic disorders. In addition, many other CS delivered, CS meaning cesarean delivered, CS delivered infants have higher risk of the colonization of antimicrobial resistant opportunic pathogens in the gut, which weakens and leads to an increased susceptibility to infectious outcomes. While the majority of studies indicate that delivery mode is the strongest factor determining early neonatal gut microbiome colonization, many other studies show that these effects are either extenuated or largely absent. In this context, several arguments have been put forward to explain these conflicting picture. First, it's important to consider that CS delivery is most often accompanied by the administration of antibiotics to mothers due to local health regulation or hospital practices. Second, colonization of the infant may be affected differently by elective versus emergency cesarean delivery, and most studies do not mention how the cesarean section was performed. Other factors, such as this gestational age and feeding practice, are also sometimes neglected. So, apart from confronting factors, the methods and study design employed over the past years may in part explain some of these conflicts, notably the frequent use of 16S RNA gene amplicon sequencing does not offer can you hear me?
Yes, we can.
So, okay. So, apart from confounding factors, the method and study designs employed over the past years may in part explain some of these conflicts, notably the use of 16S RNA gene amplicon sequencing. Okay. So, taking all these things together, we have this objective considering this project, which is the characterization first of the newborn gut microbiome in Tunisia, and also the Prevalence the Microbiome. Second, we would like to do a qualitative and quantitative comparison of the direct effect of the mode of delivery on gut microbiota diversity by avoiding, of course, the potential confounding factors that I have already mentioned. And finally, we would like to study the impact of delivery mode on the dynamic of the establishment of pioneer bacteria community on newborns, and to correlate these bacteria between and do some correlation with between the pathogens, especially bacterial pathogens, and the delivery mode. So, for instance, we here in our analysis, we have something like 33 samples from 10 newborns, 10 newborns. Okay, and we got 3 time points. The first is at the and regarding the newborns characteristic, we have all of them are healthy, and they were delivered with in a full term, higher than 37 weeks, and did not receive any antibiotic treatment, and with a gestational age of 38 weeks. And the mothers had no declared pathology, aged in mean by of aged 31 years old, and did not experience any complication during the labor and the delivery. So to address these research interests, we collected in a longitudinal manner stools samples from Tunisian newborns. We established a list of specific inclusion criteria. First, the cesarean section had to be unlabored in order to prevent the newborn from getting in contact with the vaginal flora of the mother during birth. All the mothers included in the study came from the same semi-rural region called Zahwen, located about 60 kilometers from the capital. We have only included healthy mothers who had not taken antibiotics during the last trimester of pregnancy. The collection was done in 3 steps. First, on the birthday, we collected the first tools after receiving of course, the parental consent. A series of questions was asked to the mothers in order to collect information, some data information about the diet, socio-economic situation, and health status, and so on. And then we pursued with the collection on the day 15 and the day 30 at the patient's home. A follow-up of the breastfeeding mode and the health status of the newborn was also carried out. Okay, a second step now of the project was to set up a protocol for the extraction of microbial DNA. Once this protocol was established, we proceeded with the extraction and transferred this DNA to a sequencing platform for shotgun sequencing. Why shotgun? Because we want not only to see the bacteria, but we also wanted to see other features such as viruses and also protozoans. So we selected this sequencing technique also because it can achieve a higher resolution reaching the strain level and cover all the domain of microorganisms. So the data received when then analyzed by bioinformatic pipeline and by statistical analysis. I won't show these pipelines because they are somehow not interesting for the purpose of today's meeting. Precisely, we can only say that we compare the taxonomic diversity, taxa composition, and the dynamic over time between gut microbiota for CS and VD. And we have also identified the DA taxa in both groups, DA for differentially abundant taxa in both groups. Now, we will go through the diversity analysis. So, to address the impact of delivery on these newborns gut microbiota, we carried out various taxonomic diversity analysis. Within sample diversity, as measured by alpha diversity, showed that there is no significant difference, neither in terms of evenness nor in richness between the 2 groups. Actually, the bacterial diversity between samples, as was calculated by the beta diversity, showed that the delivery mode has no impact on the global structural composition in newborns gut microbiota. Even though the prior analysis did not identify major changes in the bacterial community structure in stool sample collected in CS and BD. Smaller but very important taxonomic differences exist between the two groups. This was highlighted by linear discriminate effect size, more commonly known as LFSE analysis. Analysis showed that this analysis, which was used to identify any differentially abundant bacteria at the phylum, class, order, family, genus level. Interestingly, we observed a higher relative abundance of bacteroides that was found in children born only by vaginally delivery. This phylum is known to be immune system modulator. We can also observe that the CS newborns microbiota showed an interesting enrichment in Clostridium genus, and more specifically, the Clostridium perfringens, an opportunistic pathogen commonly found in the hospital environment. Additionally, CS showed that an overrepresentation of the Enterobacter species, a highly virulent and antibiotic-resistant member of the escaped bacterial group. This part, I will go through this part later on this talk. Okay. So, we next sought to analyze the dynamic of the establishment by comparing the three samples sampling time points. I remind you that we have 3 time points: D0, D15, and D30 after birth. This allowed us to clearly identify the temporal patterns and trends in terms of taxonomic diversity. The alpha and beta diversity analysis clearly and significantly discriminated, as you see here, the D0 in green samples from the two remaining time points, D15 and D30, okay, as shown in the both figures at your right. And this chronological pattern was similar either in CS or BD, suggesting that the colonization in the CS newborns are not delayed. And we next aim to understand how the composition and the development of developmental trajectory of the early gut microbiota occurs in CS and BD. While the gut microbiota of CS neonates was colonized by actinobacteria, proteobacteria, and firmicutes, the microbiota of BDs showed additionally the emergence of Bacteroides phylum exclusively represented by the genus Bacteroides here in red. And we also noticed that even with a lower relative abundance, the firmicutes remain constant in the CS while increasing in the BD, possibly due to the expansion of Violina genus. In addition, some genus are exclusively observed in CS, namely the Enterobacter, Rauta, and Streptococcus. And while other such Sphingomonas and Actinobacter genera are present in only BDs as shown here in pink. And finally, there are some interesting things that some genus were present in both groups but showed different dynamics in CS and BD, as observed for Escherichia and Klebsiella. This is interesting. Two species open the window of investigating the pathogenicity. So now we talk about the pathogenicity, the pathogenic prevalence according to delivery mode. So ESCAPE. This is the acronym of a very well-known group of bacteria. This group of bacteria is composed of opportunistic pathogen and related to nosocomial infections. This can cause life-threatening nosocomial infections among critically ill and immunocompromised individuals, and is characterized by potential and very well established, not potential, drug resistance mechanism. Okay, and the acronym is simply for these escape pathogens are the Enterobacter species, Staphylococcus, Pseudomonas, Enterobacterium, Klebsiella, and Actinobacter. So, among the newborns delivered by cesarean section, 84% carried opportunistic pathogen species during the neonatal period, in comparison to almost 50% of the vaginally delivered babies. During the first 3 weeks of life, these opportunistic pathogens accounted for 30% of the species level abundance in the gut microbiota of babies delivered by cesarean section, compared to only 9% in the vaginally delivered babies. In a longitudinal manner, the difference in the abundance of all opportunistic pathogens combined persisted in these babies, in babies delivered by cesarean section who were resampled later during infancy. The frequent and abundant carriage of opportunistic pathogen was also observed in vaginally delivered babies who had the lower bacteroides profile. And the absence of the breastfeeding during the neonatal period was associated also with higher carriage of Clostridium perfringens and also some Ficaris species. This disruption, the transmission of the maternal gastrointestinal bacteria through delivery by cesarean section and maternal intrapartum antibiotic prophylaxis predisposed newborns baby to colonization by clinically important and opportunistic pathogens that circulate in hospital environment. Here we can observe that further differences in bacterial disruption depending on the child being born by means of emergency or elective cesarean, as you see here in this figure. So here differences were seen in early colonization by Clostridium species. And these children born by means of emergency cesarean section differs in colonization by some other species such as Staphylococcus aureus or Staphylococcus epidermis, in which children born by means of elective cesarean section and naturally delivered were similar. No differences were observed between types of cesarean section regarding E. coli and pneumonia, whereas both different from natural birth. Okay, and we observe that DCS showed showed here showed an enrichment in terms of some genus such as Clostridium genus and more specifically the Clostridium perfringens. This is the distribution actually of the opportunistic pathogen in Tunisian newborn, and we have in higher part of the graph those that are more abundant for DCS, and the lower ones are more abundant for the delivery for the vaginal delivery. And at day 13, the CS sample showed an enrichment of Klebsiella, Oxytoca, and various Enterobacter species, as you see in here in pink. Okay, and to emphasize these aspects, we have done the distribution of opportunistic pathogen for Tunisian newborns. And as you see here, there's a very clear pattern showing that there's a difference between the both both type of deliveries. At the D0, the difference is not significant, but the more we get in time, the more this difference is seen. As you see here, we have significant difference at the day 15 and also at the day 30. So this show that it's maybe this delay of difference after the birth may be considered as the time for the bacteria to establish within this ecosystem. So we can expect also that if we can have more timing points 3 or 4 months after birth, this trend can be confirmed. So what we are expecting now for our project and the future challenge that we will go through regarding this health status of newborns and metagenomics aspect, would like to do phagiome and virome analysis regarding this metagenomic data, and we also would like to see these differences regarding also the microbiome analysis, because we know that there are many Candida albicans species that can be that are highly correlated to the declared and or higher risk of childhood asthma and many other inflammatory bowel disease that can be declared later on life. And there's a suspicion of disruption of fungal community within the newborns microbiome and to these non-communicable diseases. So to go back to the phagiome aspect, we would like to analyze the antibiotic resistance bacteria that can be that can arise in these 2 cohorts, and just to let you know that there are 10 more times bacteriophages compared to the phages, and these newborns there's an early colonization and an early colonization of these bacteriophages, and it's highly rich, and this colonization is very important within the first 4 days of life. And the difference between the phage prophage antibacterial patterns between CS and VD was frequently reported, and this aspect need to be more investigated regarding the prophage. The prophage are the bacteria that are integrated within the bacterial genomes. So these need to be more investigated and this is what we are doing right now. And we also want to see if the balance between lytic phages and activated prophages can be, if this aspect can be correlated to health status. So this part of the project, we are going to go deeply on it in these next week, sorry. And we would like also by this mean to characterize the heterogeneous newborn phagiome and to have a qualitative and quantitative comparison of the mode of delivery on the phage diversity and also prophages, and more specifically, we would like also to see the identification of genes that are encoded by phages within the 2 groups, and why not to take into account these what are the potential functions that can be brought by these genes encoded by the phages. So for this, actually, here we confirm that the metagenomic analysis is a powerful tool to decipher the prevalence of pathogenic features in various ecosystems. Actually, in Tunisia here, there's an overrepresentation of delivered newborns by cesarean section, and we have the second higher percentage of cesarean delivered. So, regarding this disruption and this important difference shown in terms of diversity and in terms of acquisition of antibiotic resistant pathogens, it's important to take this message to the stakeholders and the decision makers here in Tunisia, in order to better emphasize this important message regarding the future health status of newborn. And just to let you know that there are also potential of there's an important potential of applying the artificial intelligence algorithm and program in depicting the interplay of communities. The interplay of communities is the interactions and how these communities can interact together by quantifying numerically the abundance of each species, and how this quantity can how this interplay can impact the health status of newborns. And there are many various ready-to-use pipelines. For instance, there are many Galaxy instances with that can allow even non-bioinformatician to do such analysis. And also my favorite is also microbiome analyst that can be used to do all the kind of stuff that I have showed today. So this work was mainly done by Mariam Haneshi, PhD student, that have undertaken all this analysis on the microbiome analysis, microbiome data. And we have also Ishraq Akwili, that is a master student, that is she will be more focusing on the microbiome aspect. And I would like to thank also Ali Kahla, our head of laboratory, and here I am, the smiling guy here. So thank you all. I hope that I'm sorry for the bad connection. I hope that my presentation were good enough today. Thank you.
Thank you, Osama. We're going to take questions. We have about 10 minutes for that. So I think I will start off. I might have missed it when we were beginning the presentation. Did you mention about your sample size for this study? And also I'm curious about the difference between the elective cesarean section and the emergency cesarean section. What are some of the physiological differences in those two procedures?
Actually, we have used 33 samples. We have 10 donors, and for each donor, for each newborn donor, we have 3 times sampling time at D0, D15, and D30. And we have sequenced this with 150 per end length sequencing with the shotgun sequencing. Is that your first question, isn't it?
Yes, that is true. So do you think those 33, so the 33 is distributed between the normal vaginal delivery, the emergency, and the elective cesarean sections?
Yeah, absolutely. Actually, I will show you again. Here.
I think you had talked about 33 samples. So my guess is, since you have 3 time points times that's 50, plus the 3 replicates making 33. So I think
we have for one of the sample we have taken it twice.
Okay.
One of the babies. That's why we have 33 instead of 30. Right.
Okay.
And regarding your second question, considering the programmed cesarean delivery and it's very important to know it, because for programmed cesarean delivery, everything is prepared. Is prepared first, and there's in some cases there is no need to use the antibiotics to undertake this to undertake the delivery, because undertaking this delivery might be stressful for the mothers and also for the babies. That's why it can be sometimes it's not recommended to do it. And also for non-programmed cesarean delivery, there's in major cases the mothers were administered antibiotics before starting the delivery. That's why we decided to exclude all the emergency deliveries in the study.
So the emergency cesarean had antibiotics administered. Okay.
Yeah, because in emergency cases, we can have the real feedback or the right feedback from the doctors, because they do in some cases some things in emergency statutes, so we can evaluate our samples, our core. Sorry.
Okay. I have another question, but I'll let Tracy ask her question first. Go ahead, Tracy.
After Tracy, I'll go next before Martha, if that works.
Okay, please raise your hand if you have a question, and you can ask afterwards. Thank you. Tracy.
So I raised my hand. Yay. Very interesting presentation, Dr. Sawai. I think it was like, I had no idea that the vaginal delivery had so much impact on I don't know if it's the microbiome or whatever, which is also strange to me. I don't know if, because I would assume that the newborn or the baby's skin is the only thing in contact with the vaginal canal, but maybe you can explain that a bit more. But my question is actually, do you think that you want to use this study to inform or to make more decisions on a public health level? Because I don't know about Tunisia so much, but I know in the West, it's becoming very almost fashionable to have an elective C-section, and doctors are even advising it, because obviously it saves them time that the woman would be in labor. Do you think, or are you hoping to use this type of research to discourage elective? I mean, that's the message I kind of sort of thought I got through, but I'm just checking if that is your aim.
Okay. Thank you, Tricia, for your interesting comment. Actually, from the beginning, we were some of our deliverables is to organize a workshop with the decision makers, doctors, and many of persons of civil society, in order to discuss about this important trend. Because in Tunisia, as I've said previously, we are the second country considering the rate of cesarean delivery, and most of these cesarean delivery are not necessary, because let me be clear about this. This is done only for the sake of money, okay? Because when there's a cesarean delivery, there's more money, okay? And in some cases, unfortunately, the mothers do ask for cesarean delivery in order to avoid some contraindication during the delivery. That's why there's no, here in Tunisia, for now, there's no, people are not aware about the importance to avoid the cesarean delivery, and to let it only for the emergent cases. And our results demonstrate again that even when we have this kind of delivery, it's not without impact on the health status of the babies. But let me tell you that in many studies, they said that the newborns will have an equilibrium and balanced microbiome just after 3 years of life. So there's this trend that we observe here will disappear at the third year of life. Okay, but this is not a good idea to have this overrepresentation of cesarean delivery, especially for those cases that are not urgent.
Yeah, I'm just a follow-up, just a comment on that. It's just that I'm wondering whether the target audience here is incorrect, because I know that there were some studies in the past about how the depression rate in mothers because of not having a vaginal delivery is higher, because the mothers do not really feel that connection that would happen if there's a vaginal delivery, because of certain hormones that are being released and triggered when the baby passes through the vaginal canal. And I haven't seen any changes, even though that research has been published. So I'm wondering if maybe the target audience should be the mothers or the potential mothers, rather than the doctors, who you are saying are doing it obviously to make more money, and maybe they won't change anyway because money is more important.
Yeah,
but
yeah,
that's just my comment.
Yeah, but I think that if I ask one of the mothers and tell her that there's a risk to have the colonization by Staphylococcus aureus, one of the nosocomial very endogenous species, and that this species is very resistant and multiply and extremely drug resistant to antibiotics, I think the mothers will think twice before asking for cesarean delivery. And I hope also that this aspect will also let the doctors be more aware about this thing, even though there's a loss of money on it.
Okay, thank you.
All right. So let me quickly go. So my understanding is that the presence of the antimicrobial group does not mean that they are expressed. So my actual question is that, can you do, I'm not super familiar with the kind of analysis you're doing, but I have some experience with metagenomics. But can you do metatranscriptomics on the 16S RNA gene sequences to determine the expression level of these antimicrobial resistant groups? We know now that they're abundant in the CS delivery, but then we are not certain that they're actually, like highly functional or expressed and stuff like that. And my second question, which will just be very quick, is that, so you mentioned the application of AI in depicting interplay communities, but with the small data set you're working with, I'm a little curious how that is going to work out, unless you're going to be using some massive data sets in similar studies that, they've been able to put out their data for you to use for these machine learning task that you're working on. So those are my questions.
Okay, I will first respond to the second question. Actually, you're absolutely right. With 33 samples, it's not enough to do deep learning or machine learning aspects. But by compiling the results from all that we have concerning this question, we can have thousands of samples, and this can be a good starting point for doing artificial intelligence. And and this kind of learning can be done not only about the number of samples, but it will more focus on the number of species that are interplaying together. And this might be a good starting point to do some artificial intelligence first while doing some deep learning aspects, and can give some insights about the interplay between species. And sorry, Olaiten, your first question was about?
Yeah, thanks. I get the AI partner. Yeah, pretty much working more with it. So my first question had to do with the expression level of the antimicrobial group, because we know of the abundance, but we can't really say that they are, functional or that they are doing what they might potentially do, right? So how do we determine that with your
very interesting point. I'm absolutely aware that all that we are seeing right now, it's not about functional level. We have only insights about the genetic and the presence of some genes of some species. We are absolutely okay with that. But it will be very interesting to complement this by meta-transcriptomic, so we can have an idea about the transcription. But this is done by with very simple assumption that there's a correlation between what is what we have at the gene level and what is transcribed. So this is an assumption, and to have more clearer pictures about this aspect, doing something like meta-transcriptomic and metaproteomics would be very helpful, and will give more impact on the analysis done on the metagenomic level. Yeah. So,
okay, thank you. So you can do that also with your 16S RNA gene sequences. You can do those other analyses, the transcriptomics as well as the proteomics.
Yeah. We have to do. We have only to get some money.
Okay. Yeah. Thanks.
Thank you.
Yeah.
Thank you, James. So, Tieno, we have time for only 2 quick questions. So, James, please go ahead.
So I actually have just one question. So the question is, so you say that the differences that you observe between the CS and the vaginal delivery sort of resolve at age 3. My question is, like, now between, let's say, birth and these 3 years of age, is there some real world data about whether these differences that you see in terms of the bacterial communities actually reflect in terms of hospital readmission, you know, based on the differences in the bacteria? So do you find one particular group getting back to the hospital a lot more than the other, or seeing the doctor a lot more than the other based on the differences in the bacteria? Or are you also able to, for example, also measure antibody responses between these 2 different groups that also reflects, you know, a lot more on the physiological states rather than, you know, just the determination of the bacteria in the different groups? Thank you.
Yeah, welcome. So actually, what I said about that the equilibrium is reached again at the age of 2 or 3 years old. But it will be interesting to have more broader and more longer studies to get some results until the adult age, because nobody knows what happens between these all. What is the outcome? What is what comes with these opportunistic pathogens during the childhood, and what happens to this, and if there is an overrepresentation of this species in the adult age, and if there's any correlation between this species and some phenotypes that can be or might be observed in the adulthood. This is very important and necessitates a huge analysis with many with more longer analysis. So I hope I have responded to your question.
Yes, thank you.
Yeah.
Okay, so I have a quick one on your future plans. I think it was in the last 2 slides where you aim to investigate the antibiotic use, the microbiome, and asthma. So my question is, in these plans, sorry, the one with the baby, yes, this one.
Okay.
So, and higher risk of childhood asthma. So the microbiome rather. What other factors are you planning to incorporate in this analysis? Because, for example, rhinoviruses alone have been attributed to more than half. They've been attributed to more than half cases in childhood asthma. So I think some of these factors should be incorporated in this kind of analysis to just make sure that results will now we can investigate the role of the microbiome will come out more effectively or more accurately.
Yeah, very interesting point. Actually, what was also you asked about the rhinoviruses. but it's very important to know it. And also what is also very well established is that there's a higher childhood risk of asthma, and the prevalence of some of Candida albicans species, considering regarding the microbiome. So, and for the rhinoviruses, here we assume that the babies did not suffer of any of rhinitis, because we have we have the forms that have to be filled with the mother's newborn, in order to have any what are the diseases that suffered from which suffered the newborns. And for everyone baby that have faced any known disease, and any disease that were observed by the mother, these newborns were excluded from these samples were excluded from the study.
Okay, but I think it would be nice to just maybe incorporate, I don't know, some form of serology to check for rhinovirus antibodies, because it's a very strong factor when it comes to asthma. So it should, and children have already been shown to have the highest burden for rhinoviruses.
Yeah, listen, sure.
Okay, thank you very much. That was an exciting talk.
Thank you.
So we'll have our next pathogens course meeting in 3 months from now.